it over to David Horney, who's very active in our chapter on program and education committee. David is a 2012 graduate of MIT and in the course 2. Did I get that right? Good. He will introduce the speakers to you, and he's had a big part in getting this program together, so a big hand for David.
Thank you, Chris. So tonight it's actually the second...
Not so close to the mic.
Okay. Tonight's our second event in a series that we call a Tech Showcase. And our Tech Showcase concept is to highlight technology and products that are poised to disrupt markets. So we're talking about big ideas, sort of moonshots, that are really going to disrupt things. And tonight we're going to be discussing product that's poised to disrupt part of the drug industry, specifically antibiotics. So, but at the same time, when putting this together with Ruprecht, so Ruprecht was a co-producer of this event, and I unfortunately couldn't be here tonight, sort of realized that this is bigger than just a tech showcase. What we have for you tonight is a story of innovation. It's a story of innovation to solve a very an end-to-end problem in human health, and we're going to talk about sort of the long problems of sort of long-term problems of innovation that scientists solve, and then also the shorter-term problems of innovation when entrepreneurs want to get a product to the market to actually help the people who need it. So what we have is this rare event where essentially a scientist who's working on long-term problems. Oftentimes scientists work their whole lives to make significant contributions to problems of value, and they can solve part of it. But tonight we're lucky to have Professor Rich Gallo, who's characterized skin disease, a specific type of skin disease, sufficiently that he's able to actually take his over two decades of research and bring this and get it to the point where it's ready to be commercialized, where his insights are ready to be commercialized. So this is a remarkable and wonderful accomplishment. But then we're also going to talk about the story of innovation on the product development side and the development of precision probiotics based on the work and insights from Professor Gallo. So who's suffering from disease, how to diagnose, what's the end-to-end process for making sure the people with the problem can be helped. So this is also an exercise of asking the right questions, making hypotheses, and doing analysis, research, and pivoting if necessary to make sure we can solve this problem and finally bring it to an end. So just to put this all in context, we also invited Professor Rob Knight, whose work in the microbiome essentially is very pioneering. So he's going to lead off our discussion, and he's going to sort of set the stage for this whole microbiome problem that we're going to be focusing on tonight. So. Just want to introduce the speakers right now. So if each of you could come up as I introduce. Professor Knight, he's a professor of pediatrics and computer science at UCSD. His lab research involves the development of laboratory and computational techniques to characterize the microbiomes of humans, animals, and the environment. He's the co-founder of the American Gut Project and also the co-founder of the Earth Microbiome Project, initiated to collect data on microorganisms and ecosystems across the planet. As a result of this pioneering work in microbiome research, he's just received the 2017 Mastery Prize for outstanding contributions to biomedical sciences and the advancement of health. So please, Professor Knight, can you get us started? Thank you.
Okay, great. Thanks, David, for that very kind introduction, and thank you all for coming along. Let's hope that this works when we switch it over, as it did when we tested it. And it's a great pleasure to be here, especially because what you're going to see is the progression of the microbiome from basic science to what Rich is going to tell you about translating these discoveries into something that's commercializable, and then Mark will tell you about how it's in fact being commercialized. So you'll see a very natural progression there. And one thing to keep in mind, as I'll show you, is that better? So they told us not to stand too close to it, but all right. Okay, so one thing I hope I'll convince you of is that our microbiomes are not static, and we should not just measure them at one point, but to see how they change. So microbiome science is really changing how we see ourselves as human beings. I'd like to start by asking you to consider, just for a moment, what you saw when you looked in the mirror this morning. I saw an organism that's just 43% human, and not just because it was early and I hadn't had my coffee yet, but when we think of what makes us human, when we think at the level of cells, each of us has about 30 trillion human cells, but we have about 39 trillion microbial cells, according to the latest numbers, and so that's where that 43% human number comes from. But you might be thinking, well, wait a minute, surely we should be thinking about this at the level of our DNA. So let's think about that for a moment. Each of us has about 20,000 human genes, again, depending on what exactly you count, but the size of our microbial gene catalog ranges from about 2 to 20 million microbial genes. You might have seen today in Nature the latest of our papers from the Human Microbiome Project, refining that estimate at sites throughout the body. And so by this measure, we're at best 1% human. And there's a lot of excitement at the moment about systems biology, even systems medicine, but it's hard to do systems anything when you're neglecting 99% of that system, which is what we do when we neglect the microbial side of ourselves and all the biochemical functions that it encodes. But perhaps most excitingly for therapeutics, we're stuck with our human genomes at birth, right? We can't really change them, but our microbiomes, as I'll show you, we've changed profoundly since the day we were born. And as I won't show you, but you can look up other talks I've done about this topic, we continue to change them every day of our lives through things like the choices we make at the dinner buffet. So back in 2009, using what was then new 454 sequencing, we developed the first map of microbes across the human body. And I threatened to David that I was going to read off all of these names according to the parts of the body that they were most frequent in. I won't do that to you tonight, but I'll just assert, and Rich will take you through a few of the most important ones. But there's a lot of microbes that play important roles. But what I'm going to tell you about is more the higher level picture of how they change over the body. And even at this early stage, we could tell that the mouth was relatively stable. The skin was the least stable and perhaps the most interesting in its variation person to person and at different sites in the body, and then the gut is in between. And in the Human Microbiome Project, we built on this. So the Human Microbiome Project was one of these large-scale government-funded efforts, in this case by NIH, to build a map of the microbiome across the whole body. So we looked at up to 18 sites on the body. I should say this was a huge consortium of about 400 people around the country. Looked at 250 healthy people, so we didn't have any disease information, but we were looking at rigorously healthy subjects, sampled at up to 3 time points, and 4.5 trillion bases of DNA, so 1,500 times the size of the human genome. And we used a mixture of 16S ribosomal RNA profiling to read the name tags of all those bacteria, and shotgun metagenomics on a subset of samples to look at their functions. And so the great thing about this is we had all this data. That was also the terrible thing about it. And to illustrate this, here's the first file of data from the HMP. I'm showing you the first 0.1% of this file. There's another 17,000 just like it. And it's pretty hard to tell who lives where in the environment from this, right? Despite the fact that we're fundamentally doing ecology. Like, I bet you can't tell that's an oral sample, let alone the sequence signatures that let us determine that. And this is really a problem now, right? Because through citizen science projects like American Gut, which I run out of UCSD, and I'll tell you a little about later, and also various companies, you have people coming in to see their doctors and saying, you know, hey doc, I have some great news for you, which is I had my microbiome done, and here's a list of 1,000 species they found in my gut. Or better yet, here's a list of 1 million genes they found in my gut. And with all this data, you can tell me what's wrong with me, right? And what's your doctor going to do? Refer you to colleagues in psychiatry for being crazy enough to think that's going to help, right? But our challenge, our moonshot, is to make it not crazy anymore and to figure out how you could take that profile and integrate it with yourself over time, or integrate it with data reference frames like the HMP to actually get something out of it in the 12 beautiful minutes you have together with your physician in their office. And so to do this, we took a leaf from a very old book, specifically Darwin's work on the origin of species. And for those of you who haven't read it recently, or perhaps at all, The first edition is often considered a fairly dense read, so 502 pages, only one figure. So people who read it for the pictures are often disappointed. But I'd argue that you shouldn't be, because that one figure that Darwin had made back in 1859 was his first representation of the idea of a phylogenetic tree to organize biological life. And so Kathy Lozapone, who I recruited into my lab just when I started it as an assistant professor to work on type 3 secretion, which was the main thing our lab was working on at the time, decided that she wasn't really into that and wanted to do something with bioinformatics and sequences and trees and microbial diversity. And it turned out she did fine for herself despite completely ditching this project. She's now the third most highly cited faculty member at the University of Colorado at Denver, where she's about to get tenure. But her breakthrough was to exploit evolution and Darwin's tree of life to compare communities with this very simple idea called unifrag. So the intuition here, so these are phylogenetic trees where the branches represent molecular change in a particular sequence, and the tips represent modern sequences, and the internal nodes represent hypothetical ancestors that group those modern sequences together. And the colors represent the community that we find the modern sequences in. The intuition is if you have two unrelated communities, all the branches lead only to red tips or only to blue. None of them are shared, so 100% of the tree is unique. We define that distance as 1. Whereas in contrast, if you have two identical communities in complete sampling, what you find in the red, you find in the blue. None of the branches are unique. All are shared, shown in purple. We define that distance as 0. Then for any actual communities, you'll have some shared branches in purple, some unique branches in blue or red. That unique fraction or unifrac distance defines a metric between the communities. And so what's great about this is we combine it with Darwin's insight that there's one tree of life that connects everything. So we can take samples from anywhere, your fingertip, your armpit, your feces, the ocean outside, and we can paint them on that one universal tree of life and compare any communities labeled, say, red to yellow, red to blue, yellow to blue, take all the distances, store them in a distance matrix, and then use the whole suite of multivariate techniques and machine learning methods that acts on a distance matrix, like principal coordinates analysis to define a spatial map, or a hierarchical clustering, or numerous other techniques. And we also developed a pipeline to use next generation sequencing to do this at an unprecedented scale. And the first time we ran this in at the end of 2007 rather, the paper came out in 2008, we doubled the number of 16S ribosomal RNA sequences in GenBank, exceeding the total number that had been collected over the past 25 years by the whole community of researchers. And what we do is we tag each sample by introducing a unique barcode into the PCR primers using Golay formal error-correcting codes. We used to sequence on 454, but now we use Illumina, which is much more data, much better error rate. We developed this pipeline called CHIME, led by Greg Capareso, now a professor at NAU, where we take the raw data off the sequencer, figure out what sample each sequence comes from by its barcode, trim them, build alignments, group the sequences together, build trees, and use the tree and unifrac to get a spatial layout. And when we do this on the HMP data, we got this first overall map of the human microbiome. And so each point on this map is all the complexity of a sample read out by its DNA. And remember that 2 points are close on the map if they have similar microbes in terms of their evolutionary history, and they're far on the map if they have more dissimilar microbes. Now, if you think about all of the things that could make a difference in the microbiome sample of those healthy subjects, everything from how old they are to what time of day we collected the sample, you probably have your own ideas about what matters. And when I color this by the main factor, it's immediately obvious what's going on. So the different parts of the body are like different continents of this map with completely different microbiomes. And to highlight this, if I show you the mouth and gut of the first person in the HMP, you see they're in totally different places on this map. And it wasn't until we did the Earth Microbiome Project that we really understood how profound these differences are, because we could go out on the planet and ask what 2 points on the planet are as different from one another in their microbiomes as the mouth and the gut of this one person. So if you think of your mouth as being like a coral reef, complex mineralized structures covered with biofilms that perhaps your dentist bugs you about, the amazing fact is that your mouth is as far from your gut in its microbiome as the water in that reef is from the dirt in this prairie. So a few feet along the length of your GI tract makes as much difference to your microbes as thousands of miles across the Earth's surface. So this may lead you to wonder, well, where do we begin on this map? Where do our microbes come from in the first place? And if you have dogs or kids as I do, you likely have dark suspicions about that, all of which are true, by the way. So I can match you to your dog with high precision by a random forest model just by your microbes. But in all seriousness, how we're born has a massive impact on our first microbes. This is work we did with Gloria Dominguez-Bello, now at NYU, looking at mothers an hour before they gave birth and their babies 20 minutes after. And samples from all over babies who were delivered vaginally, shown here in pink, cluster with their mother's vaginal samples. In contrast, samples from all over babies delivered by C-section, including their mouth and gut, just cluster with skin of all of the mothers. And we think these initial differences in microbiome may be related to some of the differences in health between C-section versus vaginally delivered babies. And you may be wondering, where do you go after that? So we did this with Ruth Lay, now at the MPI, looking at one infant's microbiome over the first 2.5 years of life, just looking at the fecal microbiome, which you can see starts off in the vaginal region, which we'd expect from his delivery mode. And what you're seeing is the importance of integrating this one case with the HMP data, which is a technique put together by Yoshiki Vasquez, who's here at the meeting today. He's a very talented computer scientist in my lab. And so the question is, over 2.5 years, when we just connect the journey of the points on the map, one after the other, and each frame represents a week, how close does it come to the healthy adult state? Is the progression fast or slow? Does anything happen along the way? And when I start this going, you see sometimes massive change 1 week to the next, other times a little change. Remember, what matters primarily on this map is how far apart 2 points are. And so if you think your kid is a different person 1 week to the next, that can literally be true in terms of his microbiome, which is where most of the genes are, because it's bigger than the distance between any 2 healthy adults in the entire HMP. And coming up in just a moment is something fascinating. So what you're going to see is antibiotics for an ear infection and a massive regression of the microbiome, followed by a rapid recovery. That went by pretty fast, so I'm just going to rewind it for you and play it again. Remember, each frame is a week, so what you're seeing is that in just a few weeks, you see a massive regression of the microbiome and bouncing back to recovery again in just a few weeks. And then by the time he's at 2.5 years, you can see he's in the healthy adult microbiome. But that doesn't happen for every child who gets antibiotics, or even for every adult. And one thing we're working on at the moment is predictive models to ask what makes a microbiome stable or unstable. So we're going to switch over to the skin, and this is work with Peter Dorrestein and Rich Gallo and a number of others, characterizing the skin microbiome at unprecedented resolution. And so Peter managed to convince a male subject himself and a female subject to sample themselves at 500 sites on the body and run each of those cotton swabs through 2 kinds of mass spec to look at the molecules and sequencing to look at the microbiome, which we did. And the way to read these maps that are now spatial maps of the human body is blue means there's none of a molecule or none of a microbe, red means there's a lot. So you can see these unidentified molecules. Some are all over the body, others confined to armpits or groin or toenails or other locations. And we can use molecular networking where we we can do the same thing for the microbes as well. So you see Staphylococcus in the moist areas like the feet and the nose, like you'd expect from classical microbiology, Propionibacterium on the head and shoulders, Corynebacterium over the body. And so with the molecules, we can use molecular networking to cluster the spectra together and make a graph, and then match them up to reference standards. And you can see a whole lot of blue on this network, and that's beauty products. And amazingly, 90% of the molecules we could identify on skin came from products that the subjects had applied themselves. So we're repeating this with the Hadza hunter-gatherers who've never touched any of this stuff to see what a natural skin metabolome, a natural skin microbiome look like. But one thing that's cool is that we can overlay these spatial maps, and at the moment we're just doing it crudely, but you could imagine doing this with, say, convolutional neural nets or other explicitly spatial techniques. But even in a crude way of overlaying the maps, if you look at Propionibacterium on the head and shoulders, you can see oleic acid there and also on the hands, but it only oxidizes on the head where there's Propionibacterium and not on the hands where it isn't. And then similarly for the breakdown of palmitic acid to monoolein and monopalmitin. So what we can do is we can take that spatial localization data and then ask, can we prove causality by testing that hypothesis and incubating that microbe and the precursor molecule in the lab? When you do them separately, you get nothing. When you do them together, you get the oleic acid and its oxidized form. So that's how we can go from pattern observation to a mechanistic understanding of which microbe does what on the body. And we're trying to develop this into a high throughput pipeline for discovery of these biotransformations at the moment. We can also do this on other scales like Peter's office. So you can see a molecule lighting up me and lighting up Larry Smar, who's our director of CalIT2 very brightly, but not touching Peter at all, although there's some on his floor. And the molecule you're looking at here is caffeine, and both Larry and I drink a lot of it, but Peter doesn't. And you can see someone spilled some coffee on his floor at some point in the past. And while this might seem frivolous, you could imagine all kinds of commercial or industrial or other environments, especially hospital environments, where you really want to know where are all the molecules. And we did this for thousands of molecules and also thousands of microbes. So you can see that Synechococcus lights up Peter very brightly. It doesn't touch the rest of it. What you're seeing there, because you're probably thinking, hey, isn't that a marine microbe, is Peter swims in the sea frequently. We see the traces of it left behind on his body and on the objects he touches. One thing that's interesting about this connection between human and environmental microbiomes, you might have seen news reports about this paper that we did with Chris Larry that came out in PNAS last year. If you use the right soil microbe, you can actually vaccinate mice against an analog of PTSD. And in fact, other environmental microbes have been shown to reduce a whole range of different conditions, ranging from asthma to food allergies. And this raises a fascinating possibility that keeping ourselves too clean has cut us off from beneficial microbes we'd normally be exposed to in the environment. So as I mentioned, I launched this American Gut Project. So it uses crowdsourcing and crowdfunding to find out about microbiomes, not on the scale of a couple of hundred people like the HMP, but on the scale of over 10,000 now, because it's all supported by the participants. And so basically this allows you to claim a pin for yourself on the microbiome map. And it turns out that not everyone wants to know what's in there. So these are middle schoolers touring our lab, learning we're going to use lasers and robots to understand the bacteria in their poop, which is all literally true. But on the scale of tens of thousands of people, we're making all kinds of discoveries of things the microbiome affects that you might have had no idea about. So you might have expected it was associated with your age, which it is. But one thing it's just as statistically significantly associated with is how many hours of sleep you get at night, which you might not have expected. But it turns out that germ-free mice have no circadian rhythm, and it's only established when you colonize them with the right microbes. And anyway, on the scale, we can take this large reference set and then subsample from it to ask how many people per group you need to power your new study in the microbiome based on what we've seen before. And so these power curves show you the number of people per group versus statistical power of the test. And so you can see that for strong effects like age or IBD or antibiotic use, you needed a few dozen people per group to see those effects. But we can also see subtler effects like what sex you are, how much you sleep, how fat you are, how much you drink, how much you exercise, or I should say how much you say you exercise, because all this is self-reported data. So either that's an effect of exercise or of lying, and we're doing some more controlled experiments to find out which. But you might see that the steepest curve on this graph, so the biggest effect on your microbiome, is how many different kinds of plants you eat. And we never expected that. And what's amazing is that lifestyle factor that you have control over has a bigger effect than drugs, bigger effect than many diseases. And speaking of diseases, one of the most compelling examples we have about why you care where you are on this map is antibiotic C. difficile infection. So these are microbiomes from C. diff patients. C. diff kills 14,000 people a year. It's one of the most important hospital-acquired infections. And you can see that their gut microbiomes look nothing like a healthy gut microbiome. And so 4 of these patients are going to get a fecal transplant from this one donor. I won't show you the pictures since you just all had dinner. But basically the question is, like with the child, can you transform the microbiomes, but in this case by giving a fecal transplant from the healthy state to these 4 of these people who are sick? And so the question is the progression going to be fast or slow? Is it going to be complete? Will you even see anything at the whole ecosystem level, given the complexity of these ecosystems? And now, instead of being being a week for each frame is going to be just a day in the lives of these patients. And this is with Alex Karutz and Mike Sadowski at the University of Minnesota. What you can see is immediately, in just 2 or 3 days, their microbiomes completely transform into the healthy state from the unhealthy state where they started. And you can see through the months of follow-up, they stay in that healthy state. And in fact, the last major trial directly comparing fecal transplant to antibiotics for C. diff, which is the standard of care, had to be stopped early because the fecal transplant was over 90% effective, which is typical. The antibiotics were less than 30% effective, which is also typical. And the trial had to be stopped as a futile trial because it was unethical to continue to withhold the fecal transplant from the people getting the antibiotics. So they all got the antibiotics and most of them recovered. But the question facing us now is how can we identify these problems with the microbiome and guide them back into health? For all of these different diseases that have now been linked to the microbiome, including ones that you really might not expect, like, for example, autism has been treated successfully with fecal transplant in at least one small clinical trial. And essentially what we need to do is to find the good and the bad places on the map, and also and we're working on that across medical specialties at UCSD right at the moment. But we really need to go beyond that, not just tell you where you are on the map, but give you a microbial GPS that tells you where do you want to go and what do you need to do turn by turn to get there. And ideally, we need to develop this and make it so easy to use, because I'm in the pediatrics department, that even children can use it. And you could imagine having a smart toilet where as soon as you flush, it's going to deliver the data to your smartphone, which, let's face it, I bet you're using in there anyway, and just show you, are you going in a good or a bad direction, and what should you do? But ideally, our vision, and this is really the moonshot thing, is even more hubristic, to make it something that's as natural as examining yourself in the mirror in the bathroom rather than having to go to a lab. Now, there are already breath tests for things like cystic fibrosis exacerbation and for SIBO. So imagine if you breathed on your mirror and you were able to get an instant chemical analysis of the microbes in your breath that would use some of the same kinds of AI that backs Google Translate to be able to convert that into a microbiome profile like you get from American Gut, and ultimately put you on that microbiome map, which we derive with principal coordinates analysis from unifrac distances, highlight from your position on the map if you're at risk for any particular disorder, and then maybe give you some hints about what you should do in order to guide yourself back into health. And because we're talking about science fiction here right now, although remember the advances that took 30 years between the Cray 2 supercomputer and your smartphone, which has more processing power, that's been packed into just 10 years in DNA sequencing. You could even imagine doing something like communicating back to your smartphone so that you can use augmented reality to pick out the one yogurt out of thousands on the shelf that has the microbe you're looking for and the chemical precursors you're looking for, and then you can scan the barcode and figure out that you got the right one. And you could even imagine having the motion tracking and stuff on your phone communicating back to your mirror to show you visions of yourself 5, 10, 50 years in the future if you do the same thing every day like you did it today versus what you might look like if you did a little better on those metrics. So you laugh, but that's the kind of crazy stuff we're trying to do at the Center for Microbiome Innovation at UC San Diego that I direct with an expanding number of corporate sponsors. We have another 3 that we'll be announcing at our event on Friday, but I can't stick their logos on here yet. And over 120 faculty members at UCSD, including Rich and many others who become members of the center, and where we have a tremendous enthusiasm and capability to extend this microbiome research into all kinds of other directions that you might have had no idea it was involved in. And so I hope I've convinced you that by integrating these different levels of omics, and particularly chemical analysis with microbiome analysis, and with machine learning and with time series analysis, we're learning to not just read out, but to understand and control human microbiomes and their responses. And you'll see a lot more of that in Rich's talk. So with that, I have a lot of people to thank. I won't read this list out again, but I'd like to briefly thank a lot of collaborators on these projects, various sources of funding, the many people currently and previously in the lab who it's been an amazing privilege to work with. And I think we're holding questions for the panel. So with that, I'll just say thank you again, and looking forward to the discussion later and to the other talks. Thanks again for putting this together, David.
Thank you so much for giving us that crash course in the microbiome. I hope everybody sort of gets a bigger picture now, and as to sort of how complex this issue is and the science that needed to go into it in order to characterize the microbiome and its relationship to human function and human health. So next up, we're going to have Professor Gallo talk about a very specific aspect of the microbiome, the skin microbiome. And Professor Gallo is a professor at UC San Diego in dermatology. He's the founding chairman of dermatology at UC San Diego and the chief of dermatology at the VA San Diego healthcare system. His research accomplishments as a physician scientist, including the discovery of antimicrobial peptides in mammalian skin and establishing new links between innate immunity and skin disease such as atopic dermatitis and rosacea. If that's rosacea. Excuse me. So please, Professor Gallo.
Thank you very much, David. It's a real pleasure to be here, and always tough to follow, Rob. Thank you for giving a big picture overview of the microbiome and the significance that it quite definitely has to human health. As Rob mentioned, the size of the microbiome is really daunting, and to lead into my presentation this evening, what I'd like to give you is an ecological perspective on the way we think as physician scientists of the microbiome, and that's this theory known as the hologenome. And it's really repeating what Rob has already told you, in that our host genetic composition not only is what we inherit from our mother and father in our chromosomes, as well as our mitochondrial DNA, but our body, of course, includes the genetic material from many members of the microbial community that inhabit what is known as the hologenome. Now, this ecological perspective is something that is not at all controversial or doubted in the rest of nature. In amphibians, crustaceans, plants, it's very, very clear, and there are many examples of absolute dependence on the hologenome, certain elements of the genetic composition of the microbes that are essential for health of invertebrates. However, in humans, we always seem to be slower to accept things, and we think we're special. And it's been a while for us to really accept the fact that the microbial genetic composition is essential for our health, and we rely on it for many, perhaps surprising aspects of our biology. However, there are problems, and that is there are many microbes in our environment. Just for bacterial species alone, it's estimated that there's at least 10 million different bacterial species. That's a great underestimate when one considers that each species likely has many hundreds of strains. And most of those bacterial species we never come in contact with, probably live outside of our hologenome. And then many of them are within our hologenome, but what are their real relevance to us? Contrast that with the microbes that actually are pathogens. And as an immunologist and a physician scientist, I'm quite proud of the fact that our immune system has enabled very, very few microbes to really take advantage of us. And if you look at the totality of all human infectious diseases, there's actually less than 1,000 pathogens that have ever been described for humans. And those pathogens are specific to humans. You know, there's different pathogens that take advantage of other mammalian organisms and other types of organisms. So there's quite a ratio here of how perfect our immune system is. And we're here, and the field is exciting right now because we're trying to pick out of this vast amount of genetic material and this vast diversity of organisms, you know, do some of these microbes indeed promote health? Which ones are important? Which ones are irrelevant? And that's the kind of problem that the types of computational analysis that Rob has described is trying to solve for us, to give people like me direction to start asking specific questions in terms of cause and effect. Okay, so in medical evidence, as many of you know who are involved in the biotech community, you know, often this work begins in animal research and case series and small reports, which makes you nod and smile and wonder if it's important. But quite frankly, the only way to get to something that you truly believe are to do randomized controlled trials, which is closer to the absolute evidence when there's multiple randomized controlled trials on large-scale populations that allow meta-analysis of those trials. So what I'm going to describe for you is kind of my journey from living down here to finally trying to get up to the point where we can start proving that these hypotheses are important to human health. So I'd like to break this talk down into 4 small parts. The first is to talk a little bit about my favorite organ, the skin, and explain why I believe it's the ideal organ to study the function of the microbiome, and importantly, not just for skin disease, although that's the low-hanging fruit. Second, to describe our work that go back over 20 years on how the skin controls the composition of the microbiome. Tell you a little bit about how microbiomes can promote disease with one specific example of atopic dermatitis, and then show you a recent example of some work we're doing that is very, very promising on how we can use the microbiome to specifically promote health. Okay, so 3 reasons why skin is great and an ideal way to study the microbiome. First, of course, you just have access to it. It's right there. Other methods for microbiome assessment, such as Rob showed you with that child on the toilet, are helpful, but frankly, it's rather indirect. Here we have the epithelial surface that the microbe is interacting with. It's right there. We can get it easily. Problem is, there's a lot of different types of skin, a lot of different surfaces, so it gets complicated. The next aspect of why skin is ideal for studying the microbiome should have been obvious a long time ago, but it wasn't. And that is that it's actually the largest organ and the largest surface area for microbial communication. If you look on your cell phone right now in Wikipedia, what's the surface area of the skin, you're going to get an answer that says like 2 meters squared, which is pretty big. It's the size of a badminton court or something like that they always talk about. But it's a dramatic underestimate that's based on the fact that topical application of drugs and so forth look at the barrier of the skin as just a surface. But the reality of it is that the skin, if you look at any area of your skin right now, you'll see multiple little holes. Those are follicles or appendages. Many of them have hair. Some of us have much fewer hair than others. But in all cases, they're deep little pits. And when you look in those little pits, of which there's at least 5 million on the average size adult human, that is the highest density of microbes. And if you look closer in that little pit, this organism or this organ is designed specifically to entrap and protect microbes from the outside world. So why do we have so many holes on ourselves? In addition to cooling ourselves and moisturizing ourselves and occasionally putting out a random hair, I think it's really there to gather the microbiome and protect it. We have specific organs that are designed to produce hydrophobic materials, lipids that trap it. So washing, soap, scrubbing, things that you want to try to do to change the microbiome on the skin do not work. And there's lots of studies now that have shown that all kinds of topical interventions do not change the microbiome because the hair follicle and this giant surface area of this bulb is designed to protect them. And if you look at that bulb carefully, you see a couple of other really remarkable things. First of all, each of these bulbs is lined by a tremendous neural network. Why are all these sensory and afferent nerves there? So it's a perfect way for the microbiome to affect autism, for example, or other neurological diseases. But the skin is not really looked at because it's not been appreciated. Another thing about each of these hair follicles is that there's a tremendous amount of vascularization. Again, the surface area here for absorption of external material is much, much larger than the gut or even the lung. Yet it's ignored because it's protected by these lipids. The only thing that's allowed to interact with that kind of vasculature is the skin follicular microbiome. And because of these kinds of observations, we're doing some sort of moonshot experiments now to try to influence diseases like diabetes by delivering insulin through skin microbiome to the organism. So there's lots of crazy things you can start thinking about systemic health independent of skin disease through the skin microbiome. Okay, so next step, skin controls the composition of the microbiome. This is the stuff I've been doing for a long time. Quite a while ago, while a postdoc at Harvard, we somewhat accidentally discovered that mammalian skin makes antibiotics. Just like bread mold makes penicillin, turns out we all make our own antibiotics. Those antibiotics were thought to be evolutionarily ancient at the time, and through a number of experiments we and others have done, we've shown these are absolutely essential for protecting us against infection, but they also are shaping the microbiome. And this is not only true for the skin, it's true for every epithelial surface. It's a very complicated system, but there's some simple experiments that I hope will convince you that these are important. And this is the experiment I love to show, which you can do in your garage if you like. And it's a simple experiment where you take a bacterium like Staph aureus that has evolved to be a skin pathogen. This is a bug that likes skin. And you can take a bug like E. coli, you've all heard of that as well. This is a bug that likes the gut. And if you wash your fingers, and this is 3 fingertips, and touch them to Staph aureus or touch them to E. coli and wait 10 minutes and then go back and touch an agar plate, the agar plate the next day will grow little colonies telling you if the bacteria lived once it touched the surface of your skin. And it's very obvious here that the Staph aureus, which has learned to evade some of the defenses of your skin, survives, but E. coli, which is such a terrible pathogen when ingested, is immediately killed, and your skin surface is basically sterilized by your innate antimicrobial defense. And every experiment needs a control, and the control for this experiment, I think, is even more impressive, and that is, what if you did the same thing, and instead of having bare, washed hands, you put on sterile surgical gloves? Well, now, if you take your surgical gloves and touch Staph aureus, or take your surgical gloves and touch E. coli and wait 10 minutes and go back, look what happens. Actually, the Staph aureus can't even survive so well in the glove. It needs the skin surface's nutrients to survive. But the E. coli goes to town. It goes wild. It's very happy. So what does this prove? Well, I'm showing it to you because I want you to believe me when I say that the skin surface is the epithelial surface is shaped the microbiome. But I also want to show you that when you wear gloves, you're protecting bacteria from you. And when your food service person is wearing gloves behind the counter and serving you their mashed potatoes, what they're really enabling is the transmission of E. coli. Sorry about that. The system is complex. As I said, there are many dozens of antimicrobial peptides and other components that contribute to this. The cell types that do it are very specialized. Even fat is important in protecting against infection, as we showed in a science paper a couple of years ago. And other surprising molecules like microRNAs can also help shape the microbiome. So we expend a tremendous amount of genetic energy in our body to have that microbiome be just so. And it's different for individuals, and it's very different between species. Okay, some members of the skin microbiome promote disease. Well, the germ theory of disease long ago showed us they could cause infection, but we're coming to appreciate now that there are many microbes on the surface of our skin and at other organs that can cause disease that aren't classically thought of as infectious diseases. And the best example I can give you of that is this disease called atopic dermatitis. It's extremely common. There's thought to be at least 132 million people worldwide affected by the disease. At its extreme form, it can be fatal from erythroderma or superinfections that occur when you have this. It's very common in the pediatric age group, so it has a tremendous economic impact, not only because of the uncomfortableness of the patient, but how it completely disrupts families because nobody can sleep and everybody's crying and itching and scratching. And based on the prevalence of this, I would hazard to guess there's some members of our audience right now that even have this disease or have a child with this disease. And Mark will tell you more about the economic impact and some of the effectiveness of some of the recent forays into biologics for treating this disease. I'm part of a group called the Atopic Dermatitis Research Network, which was founded by an act of Congress about 15 years ago as part of the Bioterrorism Defense Fund, because the reason why we couldn't give soldiers smallpox vaccine at the beginning of the Iraq wars was that if you had atopic dermatitis, the smallpox vaccine could be potentially fatal, or you could actually bring that live virus home and infect children with atopic dermatitis with the vaccinia virus, which is, although inert in normal people, is fatal in atopics. So the government has been very interested in understanding how atopic dermatitis works and why it causes disease. And this is a study that we've done in a network on several thousand individuals, broken down here by race, showing that one of the predetermining factors for severity of the disease is if your microbiome contains a lot of one particular organism, which I've already told you about, called Staph aureus. So this is a scale of severity. If you're up here, you're super itchy and oozing all over. Down here, you're just a little bit red. And people with Staph aureus on their skin, whether they be African Americans, or European Americans, Mexican Americans, or other in our cohort, always have a more severe disease if they have Staph aureus. Staph aureus can also directly be used to cause disease like atopic dermatitis in mice. So before you cause any kind of infection in mice, if you put Staph aureus on them, they become itchy. It induces a rash that looks like atopic dermatitis, and you get a very characteristic immunological signature that includes elevated IL-4, which is the target of the new Sanofi Regeneron drug that was recently approved by the FDA this year. That's been very, very effective, the first biologic for treating atopic dermatitis. It's just targeting IL-4, and you get elevated things like IgE and TSLP and increased T cells. So you really get a very excellent model of atopic dermatitis in mice, just caused by applying Staph aureus and breaking the barrier down. And in fact, when we go in the lab and we take mice, and if you just put nothing on their skin and compare it to putting a wild type, sort of a Staph aureus from an atopic individual, you can see very readily the rash. And if you look under the microscope, the skin looks like atopic dermatitis skin. And we and others have made some progress recently in understanding how they do that, and can now target and specifically delete genes from Staph aureus that cause that disease, and that's what this Staph SA mutant means. So you can make atopic dermatitis, and you can make Staph aureus not make atopic dermatitis. Also important for this argument that Staph aureus drives the disease are some recent papers that have just come out in the last few months. This is a large-scale study done in the Netherlands in which they looked at over 3,000 patients initially and followed them from birth and looked at the ones that got atopic dermatitis. And they had about 600 patients that got the disease. And what they were able to show is that the patients that got the disease, before they got disease, they were colonized by Staph aureus. And also of interest is when they looked at the patients that didn't get atopic dermatitis, they were colonized by another member of the skin microbiome, a coagulase-negative staphylococcus called Staph hominis, which I'll talk a little bit about later. So getting Staph aureus bad, if you get it, you get AD. Staph hominis good. Other labs, other groups here from the NIH and Ireland also found similar approaches using different techniques of DNA sequencing of the microorganisms. So the overall model for this disease is that all of us who are healthy right now on the skin have a normal community of the microbiome, which is not completely defined. You have a normal skin barrier, you have your normal antimicrobial peptides, and your immune response is balanced and normal. A number of genetic defects, environmental defects, dry skin, using the wrong soap, things that break down the barrier can throw this out of whack. If you then have a dysbiosis of this community of bacteria on your skin, and it gets populated by Staph aureus then drives something that makes a rash, degrades protein, digests the skin, causes this characteristic IL-4 immune response. That in turn feeds back, decreases your antimicrobial peptides. We published this about 15 years ago in New England Journal of Medicine. And when that happens, that promotes the dysbiosis. So you get this vicious cycle, hence chronic disease. So that's the model for why we think atopic dermatitis is so common. So many different things can trigger off the cycle. Okay, so can we exploit that model with our understanding of the microbiome and actually make a difference for these people? That's the thing I care about. We and others in the laboratory have found many things that the microbial communities can do that seem to possibly be good. That includes enhancing your antimicrobial peptides, changing the balance of T cells and dendritic cells in your skin. And for our particular purposes tonight, I want to talk about one aspect of what skin-resident microbial communities might do, and that's directly inhibit pathogen growth. Could one bacteria that's normally on your skin kill another and help protect you? So this is a study based on a high-throughput functional screen that we developed with the Atopic Dermatitis Research Network, where we looked at thousands of colonies from many individuals across the country, and we assayed them for if one bacteria could kill another. And the bacteria we looked at was coagulase-negative Staphylococcus. It's many different species, and we just wanted to see, could those coagulase-negative Staphylococci kill Staph aureus? And the results blew me away, quite frankly. In the healthy population, almost every one of the 10,000 or so colonies that we isolated from these individuals could inhibit Staph aureus. So we get close to, many people, 100% reading. But in the atopic patients, even if they didn't have disease on the skin, they had deficiency in the function of their antimicrobials. So we found two things wrong now with atopics. Not only did they have a deficiency in antimicrobials from the cells, but also their microbiome wasn't making antimicrobials. And if we analyze these clones, all of a sudden we start seeing these different specific bacteria making antibiotics that we had no idea existed. These are antibiotics that you all have. Many of these, 60% of the population have. There's a vast array of them. This is really the basis of intellectual property used for developing this as a potential drug. And we've got lots of different antimicrobials, natural antibiotics, produced by human strains of bacteria living on our skin that have potency similar to pharmaceutical antibiotics. In fact, if you take a mouse and give it that AD model and put some of these bacteria, in this case just an extract from the bacteria on the mouse, you can make the disease go away, and you can show that that IL-4, that key cytokine, goes way down from just applying onto the mouse this kind of transplant. But remember, we need real evidence. We need clinical evidence in humans, so we needed to do a randomized controlled trial. In an academic lab, this isn't easy because we had to go to the FDA and get an IND, but we powered through it. And this trial design was quite simple. We figured the FDA would be least afraid if we used a subject's own bacteria. So we swabbed an individual. We put their bacteria through this high throughput screen. We found in atopics that very rare bacteria that would possibly be good, but it was suppressed in that disease. We grew it up, put it in a jar, mixed it with some cream, then did it randomized and double blind, and then reapplied it to the individual, one side getting vehicle, one side getting the cream. Just did it once. And looked the next day. And this study too, we had to break early because it worked in 100% of the individuals. Everyone who got the cream, boom, the Staph aureus just completely dropped off the map in a randomized double blind way. So that was exciting, very small study, of course. I got excited about it. Francis Gollins got excited about it. He blogged about it. The New York Times kind of depressed me because the emails get filled from New York Times articles. And even this guy, Bill Gates, came and wanted to hear about it. So that was sort of fun for me. So I think we're onto something here. I believe we've got something that has great potential that needs development. This type of strategy of functionally characterizing the microbiome can only be made possible by better understanding what the community looks like through the type of work that Rob is doing, extending this to lots of other diseases, Parkinsonism, baldness, acne, diabetes is perfectly reasonable if you develop the appropriate physical screens and understanding the molecular basis of doing it. So I think the skin is showing, for somewhat, perhaps the first time, true function and causation for the microbiome. There's immediate opportunity for characterization here that I think Mark is going to talk about. And I think there's lots of therapeutic targets. And with that, I'll shut up. Thank the people who make it possible, the department around me that keep me standing, and the NIH who allow me to pay the bills. Thanks very much.
So thank you very much, Professor Gallo. So the question is, how do you take all this fascinating science and understanding about these good bugs and actually put it into a product, a drug, and actually deliver it to people who need it? So we have Mark Wilson to talk about that. He's leading the commercialization effort of these of the translation from this microbial science into drugs, and he's the co-founder along with Professor Gallo of. And he's also the CEO of Matrices Biosciences, a clinical stage company focused on commercializing therapeutics at the interface of the innate immune system and the human skin microbiome. Mark has almost 30 years of biopharmaceutical and diagnostic experience with extensive accomplishments in pharmaceutical licensing and business development, new organization startups, strategic alliance formation with big pharma and big biotech firms. So please, Mark, welcome. And looking forward to hearing about this story.
Thank you. The length right here. So thank you very much. We are therapeutics at the interface of the human microbiome and the innate immune system. You've already seen this team, but it's important. This is Rich Gallo's team from about a year ago. The patent that we license was discovered by Rich Gallo and Teru Nakatsuji. We figured they began on the beach, so we'd stay down there. And we are still down there. We're just a couple blocks south of that, at the Scripps Research Hospital building, the original Scripps Research Hospital building. A little bit where we came from. Some of you were interested in kind of our story. In my opinion, it begins with Connect. Connect was actually founded, many of you may not know, from UCSD. So Connect was in UCSD in the beginning. It eventually spun out. I would encourage all of you entrepreneurs and all of you mentors to get involved with Connect. It's a good organization. It's a good mentoring group. Springboard program is very good for young entrepreneurs. I've been through that. I have coached companies. It's a very important group. I've been involved for, 15 years. In the 90s was the advent of high throughput screening technologies. That was very important. But in 94, Rich described it briefly, with the discovery of how the skin's innate immune system protects ourselves. So the skin makes antimicrobial peptides. That was a huge discovery by Rich Gallo and his group. In the 2000s was next generation sequencing. I think we have some Illumina people here. Thank God for you guys, did a very good job, and you guys survived. And it's a very important technology that's used today. In 2009 was the Human Microbiome Project, funded by the NIH. But at the same time was really Rich's discovery of beneficial skin microbes that make antimicrobial peptides. So that was in 2009. We were founded in 2015. So we were founded by a group that's sitting here today. Gilbert Keller, Lou Bookbinder, raise your hands, and Rich Gallo. Okay, so we spent about 6 months reviewing IP. This is really hard work. We went through a lot of the UCSD IP, a lot of it from the Gallo lab. In the end, we licensed a provisional patent from UCSD, and it was called Hogacidin, and I'll talk a little bit more about that. But it was called Hogacidin, and then we did fundraising, including the Tech Coast Angels, and eventually established a laboratory in La Jolla. So we are in the original Scripps Research Building in La Jolla, and we moved there in 2015. Our opportunity is to develop therapies for the top 5 dermatology and skin health conditions. And those are atopic dermatitis, psoriasis, rosacea, acne, and skin infections. And more than 230 million people suffer from just those 5 conditions. There are 1,300 dermatology companies worldwide that do not have an effective solution. We intend to partner with the largest of those. And just these 5 skin diseases alone are a 13 billion prescription market and a 120 billion non-prescription skincare market. So these are very big markets, but importantly, these have a big impact on the quality of life. This slide is only to show you our first product is in atopic dermatitis. The atopic dermatitis, the more broad condition would be called eczema, and the eczema market is roughly 3.8 billion. But today, the atopic dermatitis therapeutic market is 800 million worldwide. As Rich said, 132 million patients. This market is about to get much larger as biologics are entering this market. And so this is the science Rich described earlier. I think it's important to just tell that this is the innate immune system and how your body's skin cells make antimicrobial peptides. That was Rich's first discovery, and this is a very, very important discovery. But there are some times that you are colonized by really difficult pathogens like Staph aureus. And in this case, your body's antimicrobial peptides cannot fight off that infection easily. And so our intervention is a specific strain of Staph hominis. That good bug makes antimicrobial peptides that kill Staph aureus, even kills MRSA topically on your skin. This is taxonomy. Sorry, I'm not the academic, but I think it is important. Taxonomy is naming and classification of organisms. And so we start with life, and we keep going down in our breakup down to genus and species. And what we believe is you have to keep going down the march, and you have to get to the strain level, because we know that in individual species, there are active and there are inactive strains. So we are commercializing specific active strains down at the strain level. One we'll talk about more today is the Staph hominis A9 strain. But it's important here because what you can do with your microbe is you can do a genetic sequence of that microbe, and you can blast that against a database. So that's the big data part of this presentation. MIT wanted us to talk about that a little bit. And by blasting that against a database, you can begin to identify the antimicrobial peptide gene systems found in that microbe. You can see what that bug might be making. It's a good first step to see what's actually happening in that specific strain of that microbe. These are some of the known antibiotic peptides, and I put these out there to show that our logo is twofold. It is a sword and is a shield. This is the sword. This is the killer. This is a very targeted thing. These are some of the known ones. These are the ones that are new. I believe, Rich, these are the correct structures, hopefully. The first product is, the patent was called Hogocidin, and that's from Japanese Hogo, which is protection. And these are the two peptides made by what we believe will be our first product for atopic dermatitis. This is the structure of what these peptides look like. This is the lanthionine bond. These are called lantibiotic peptides. This is our pipeline of our prescription products. This is the first one that we're talking about right now. MSB-01 is an anhydrous formulation of this specific A9 strain of Staph hominis, okay? So this product is, we call it entering phase 2. We have other microbiome solutions for other indications. These are small molecule programs. These are 505(b)(2)s from Rich's earlier work. These are a cosmetic actives development pipeline. These are extracts. We have a very robust business that we believe is coming in these extracts for cosmetic uses and skincare uses. These would be non-prescription. This is a lantibiotic containing extract that could be useful for eczema, cracked skin, and that kind of thing. This is a anti-inflammatory substance, comes from the wall of the good microbe. And this is a new one. This is called Firmicidin. This may be a prescription product. This may be an extract for a cosmetic active. We don't know yet. This is very interesting. This special microbe makes a small molecule that inhibits DNA replication, so it could be useful for photoaging or UV. It could be in a pharma use for actinic keratosis, maybe even skin cancer. So this is a slide from Leerink Investment Bank. Dermatology has a lot of unmet need today, and that is being filled by biologics. These are very expensive products. Some have been approved. They are effective. Generally, they are immune suppressants, so you have a downside with that, but these are products that have been shown to be effective. This is our IP today. Shout out to Dan Yarborough from Kenobi. And Kenobi is our counsel, and they prepared this PCT. We licensed a provisional patent from UCSD, enlisted Kenobi to write a good and robust PCT, which was filed in May of last year. It includes 13 strains, different uses, different things in that. What Matrices is doing is expanding that IP through process IP and formulation IP. Sorry for these pictures, but this is atopic dermatitis again. What is atopic dermatitis? It's an overgrowth of the harmful bacteria Staph aureus and the inflammatory response. That's what creates the characteristic lesions of atopic dermatitis. And this cracked skin can also allow entry of other bacteria, even sepsis, even can cause death. This development candidate has potent anti-staph activity. It also has activity against strep A and B, even MRSA on your skin. This is a little bit complicated slide, but it's not. This is atopic dermatitis and looking at the broad category of products in that class. And the first line is generally steroids, and steroids are useful for anti-inflammatory effects, but everyone knows you can't use steroids chronically. They cause skin thinning and other side effects, but they are widely used. The second line of defense is topical calcineurin inhibitors, TCIs. These products have a black box warning because of safety questions. And they have not done well in the market, but they are topical, that's a benefit, but they are also immunosuppressants, which is not good. People also take NSAIDs topically or locally. or systemically to help with the pain, but they don't do anything with the rash. They don't do anything with the chronic 24/7 itch. There is a very good product that is approved in the market that is just getting launched from Regeneron Sanofi. This product is called Dupixent. Dupixent is an IL-4 and an IL-13 blocker. It's approved, and it's definitely has a very high forecast. It's forecast up to $5 billion today, but it's important that this is approved for atopic dermatitis that is inadequately responsive to a topical product. We're developing topical products. It is our hope and our wish that we'll be first in line for atopic dermatitis. Johnson & Johnson is also very interesting in this market. They have a barrier cream business. So these are highly emollient formulations like Aveeno that are beneficial for your skin. So what Matrices is doing is combining the very targeted benefit of this microbe that only takes out Staph aureus, doesn't take out the other good microbes, with the benefits of a highly emollient formulation as a topical product. It's a natural microbe. This is not genetically engineered, and we believe that this has the potential to be the first in class for atopic dermatitis. This is another slide from Lyrinc. This is an interesting slide because these are the two recently approved products for atopic dermatitis. These are both systemic products. This is the Pfizer product. This is an oral product. This is the Dupixent product that I just mentioned. Lyrinc says it's almost $6 billion. This is for moderate to severe AD. Pfizer's product is mild to moderate. Based on our safety profile, that's early admittedly, but based on the effects, we believe that we can target mild, moderate, and severe AD, even children, and that's a very robust opportunity, I believe. What Matrices does is we are a product development organization. So we stabilize the strain and produce a formulation that's stable and commercially viable and carry these projects through to commercial proof of concept. It's important from the partner's point of view. These are novel therapeutic products with IP in very large markets. But more importantly, for the patient's point of view, they have an unmet need here. These people need help. This is a 24/7 itch. This is definitely a patient that wants something here. But we are developing topical, non-systemic products. This is not a steroid approach that might cause skin thinning, and this antimicrobial peptide approach should not lead to resistivity. This is the go-to-market. This is very important. These are the clinical trials that are planned, and these are NIH-sponsored, but these are NIH-sponsored, in this case, to Rich's lab directly. In this case, they are sponsored to the ADRN group that Rich is one of the leaders in. So they're not sponsored to Matrices, they're sponsored to Rich and the ADRN team. But each one of these has very important clinical trial readouts that lead to our various partnering discussions that we have underway today. What Matrices is doing is a traditional biopharma development program. So we've made a master cell bank, a working cell bank of this microbe. We're carrying through engineering batches, eventually leading to GMP clinical supplies for a clinical trial in about a year from now. So this trial has just begun in Denver, also at UCSD. This is a frozen preparation of this specific strain, Staph hominis A9. The commercializable product will be trialed in about a year. So this is a topical preparation. It's an anhydrous oil formulation of that specific microbe. This is the clinical hypothesis. So when Rich's paper, seminal paper published, Francis Collins himself put in his blog how important the paper was, and he referenced this paper, the Kong paper, that talks about the temporal shifts in the microbiome. And what you have here in my world is a battle of two armies. The good guys, microbial diversity, these are the good bugs, and Staph aureus, which is the bad bug. So when Staph aureus is rising, that's what initiates the flare of atopic dermatitis. So our goal in this program is to basically defeat Staph aureus, allow the microbial diversity to again return, and eliminate the flare. This is what we're doing in the lab. Here on the left side here are various strains of this one strain, Staph hominis, in a culture. This is how it's brewed. This is a 6-liter bioreactor in the lab. This is what you might call a drug substance. This is the lyophilized milled strain of SHA9, Staph hominis A9. So this is a traditional drug substance, but it's really a milled microbe strain. These are different put-ups that we're working with. This is how they look in a product. And what's important is you can drop the lyophilized bacteria onto a Staph aureus culture. That bacteria will come back to life and kill Staph aureus. And you see that by a zone of inhibition. The formulated product, which is the lyophilized milled bacteria in an anhydrous oil, does the same thing. So we call that product proof of concept. You can make a stabilized product that's commercially viable. That's what we're here to do. We can partner, so the technology is very interesting. What we have today, there's a lot of opportunities to make partnerships with larger commercial organizations. And if you look at the comparables, these are valuable opportunities, these are valuable indications, there are a lot of people involved, and these are the kind of deals that we hope to make. We are a C corporation, so the exit for a C corporation either to go public or to be sold. And you can see that looking at comps, these tend to be valuable organizations. And just looking at the deals, as I just mentioned, these atopic dermatitis, flaky skin deal, which would be eczema, cosmetics, sinus infection driven by Staph aureus and rosacea, these are all example companies, but this is the kind of layout of deals that we hope to be making as we grow and progress at Matrices. So today, the phase 2 trial just began. I want to be very clear that that is NIH sponsored to the Atopic Dermatitis Research Network. Okay, that's not sponsored to us, that's sponsored to Rich's group and the ADRN. And this is a frozen product today, but it's a very important trial. MSB-01, the lyophilized stabilized formulation, is in full development. Master cell bank, working cell bank have been completed. Engineering runs. We are in scale up today. The product proof of concept I just mentioned has been completed. That means the lyophilized microbe wakes up in the presence of water, makes its antimicrobial peptide, and kills Staph aureus. Formulation optimization of that is underway. We have a lot of licensing discussions underway, both by immunology and dermatology companies, equally strong interest in cosmetic extracts. We've got term sheets for that side, and fundraising is underway. I want to go back for just a brief moment. Our partners are interested in partnering with us, but they want to see some of this clinical data. So that will evolve over time. That's why these partnering discussions are a continuum. And finally, we believe that we have the potential first-in-class solution to atopic dermatitis, thanks to the IP discovered by Rich Gallo. It's an $800 million market today, but this market will be growing dramatically with the advent of some of the biologics coming to the market. We're the most clinically advanced skin microbiome company, potentially first to market with the best-of-class product. Patent was filed May 5th of last year. It has 13 strains and uses of that, and we have Rich Gallo as our scientific advisor. One last comment is that there's a reason we have this logo. This is the sword. The sword is here because what we do with these antibiotic peptides, we're only taking out the bad actor. There's no friendly fire here. We're taking out the bad guy, not trying to wipe bugs off the earth. You can't do that. The other part of it is the shield. The shield has to do with the good microbes that protect us and your skin. Your good microbes and your skin are the first line of defense. Thank you.
Mark, thanks so much for that. If we could, we're going to start the Q&A now. So if the speakers could come up to the main bench here, table. So guys, thanks so much. That was a fascinating story of how a new organic drug, naturally made in the human body, is going to come to market and disrupt this pharmaceutical antibiotics. So this is something that's going to be beneficial for everybody, and hopefully this will also transfer into other new drug technologies as well, naturally occurring products and organic. So this is really fascinating. So I'll start the questioning off with the first question, then I'll turn it over to the audience here. I'm sure you guys have a bunch of questions. So my first question is for Professor Gallo. So as a scientist myself, a young scientist, you always wonder, okay, when do I have enough to actually publish something? When is it new enough? This is sort of a question for all PhD students. So I'm going to ask you, when did you realize you had enough to make this product? And then can you tell us a little bit about what your next steps were, what you did to sort of, verify and then go forth to product development?
That's a very easy question because you've never had enough, okay? It's just you've run out of time. And, grants have cycles and so forth. So at some point, you really need to, think that your work is valuable enough to publish. The product development thing, that is a team approach. It came not only from me thinking maybe it is beneficial, from Mark's group coming up after we proposed the IP to the Tech Transfer Office and wanting to license it, and other colleagues kind of coming up and encouraging us to do it. So at times I regret it because some of the product development and, we're doing GMP manufacture in our lab right now, which I had a full hair that was as dark as yours a year ago. I swear. It's just hell. And I want to stop doing that part and get back to the basic research. But the answer is you never have enough. It's always fun to do it.
Great. So it's great to have a good team advising you and saying, let's do it. It's time. So I'd like to transfer this over to the audience. Anita and Chris have mics. So if you have a question, please raise your hand. Jerry.
Hi, I'm Jerry Feidelson. Rob, I had a question about the complexity beyond just bacterial. You did say microbial, not bacterial, and I assume that's because there's some complexity introduced by protozoans and viruses. To what extent do those non-bacterial microbes complicate your analysis?
Yeah, good question. So what I showed you was mostly bacteria and archaea. When we do specialized primers or when we do shotgun metagenomics, we pick up the microbial eukaryotes and the phages as well. It depends a lot on what process you're looking at. So, for example, we've done some work with Jeff Gordon's lab looking at personalized culture collections, where what you can do is really prove causality by taking hundreds of strains out of an individual stool sample, growing them azenically, mixing them together, putting them back in the mouse, and showing that you have the same phenotype as when you transfer the whole stool. And so that's how we can prove that it's the bacteria doing the job in that system, not a virus, not a fungus, not a small molecule metabolite, not a cytokine or whatever, right? But that's not going to be true for every phenotype. In terms of looking at the coevolution of the dynamics, no one's really done a good job of that in the gut. Jed Furman at USC has a nice paper in the ISME journal that just came out this year in seawater, showing that you have to go all the way down to single nucleotide resolution to see associations between viruses and bacterial strains, and that when you aggregate at higher taxonomic levels, it doesn't work. That's probably true in the gut as well, but no one's used the technique yet there, although it'll be fascinating to try that out on some of the longitudinal series we have.
Okay, thanks. The other question I had was about prebiotics. There's a lot of marketing out there for inulins and galacto-oligosaccharides. To what extent do you think that's a scam, or how much is it real?
Well, so I used an analogy from probiotics, which is when I moved to California at the end of 2014, as one does, I decided to buy a couple of citrus trees for my patio. And right there in the gardening shop, the bag of special citrus fertilizer had in huge letters on it, new with probiotics. And I think that might have been the only time that the probiotics were literal BS. But I think metaphorically there's a lot of that going around. And the same sort of thing's true in the prebiotic space. So just because you can feed a compound to a microbe in vitro and see a particular reaction taking place doesn't mean that's necessarily what's going to happen in your gut. Now, on the other hand, the data on things like fiber and plant secondary metabolites and so on in the context of whole foods is pretty compelling. So a lot of the question is about what can you really get out of an extract. and do the changes correlate with modifications of phenotype that have any kind of clinical benefit. And if you're going the supplement route, you're very strongly motivated not to prove any clinical benefit, because then you'll be re-regulated as a drug, and you'll have to you'll have to do everything under GMP in order to bring it back to market, as well as doing about $1 billion worth of clinical trials. So it's a bit like sports supplements, where the way you can prove a sports supplement doesn't work is it hasn't been banned by WADA and the other anti-doping agencies.
Okay, David?
Just to add, as a follow-on to that, what about more macro creatures, nematodes, parasites, roundworms, that kind of thing?
Yeah, good question. So we've done a lot of 18S ribosomal RNA sequencing based on parasite detection, and in the US population, those organisms are not very prevalent, and you pick them up very seldom. In other populations we've looked at, especially rural populations in South America and in Africa, we find much more interesting diversity there. In terms of good longitudinal studies looking at the covariation of parasites with the with the microbiome, either on evolutionary scales or within the lifetime of one individual, there's been very little done, in part because of the challenges of doing longitudinal studies, especially with densely sampled time points in those populations. So one really compelling need is better preservation methods, so you can bring the samples back without having to lug around liquid nitrogen dewars, which is what we've done everywhere from working with the Hadza to the Yanomami to various other populations.
I'll just add to that. In the skin space, the Demodex follicularum is an incredibly abundant parasite that lives on normal skin. It's almost never sampled by skin swab techniques. So the technique of collection is by skin swab techniques. So the only way you know that there's Demodex there is either you do a biopsy, where almost every follicle you'll see the parasite in there, or if you do biopsy strips and pull the little plugs out of the follicles, and then the Demodex is there. So the sampling technique, in addition to the transportation and everything, makes all the difference on the results you're going to get.
And that's a really good point. Like, if you're doing a swab to collect stool, you're certainly not going to pick up a tapeworm.
Other questions? We got in the back, Bish.
So if you look at the landscape of microbiomes, so there was a good case study of atopic dermatitis being tackled. What else is out there that is equally promising, and are there things that have not been addressed yet that you think have a lot of promise using the research on microbiomes?
I'll start with that, and I'll start with skin diseases because that's what we focus on. But the next sort of low-hanging fruit is acne as a common disease, and then there's a rare genetic disease that's a protease deficiency called Netherton syndrome that also very clearly has a defect in the microbiome, and that may be driving the disease. So in a lot of these cases of dysbiosis where there's an opportunity to interfere and interrupt the dysbiosis, I think poses same with the C. diff infection, that is a dysbiosis of the gut. So the opportunity to interfere with that and correct it gives you a therapeutic opportunity.
Yeah, and it's really hard to tell what's not promising. So, for example, on the AGA newsletter, those of you who subscribe to it probably saw last week the 2 case reports going around of individuals who had been cured of alopecia by a fecal transplant. And that's certainly something you might not have predicted was linked to the gut microbiome. So it's really hard to rule things out. And like cystic fibrosis is everyone's classic example of an inherited genetic disease. But the difference between living to age 2, which was typical a few decades ago, versus age 42, which is typical for a CF patient now, essentially all of that difference is figuring out how to manage the complex polymicrobial infection. So better understanding of the microbiome can have very dramatic differences in phenotype, even if the underlying basis is at least in part human genetics or a well-understood environmental factor or other things like that. And one of my favorite examples of that, one of my favorite examples of that actually is consumption, which was one of the classic Victorian examples of inherited disease. So, like at the time in 19th century London, everyone was exposed to Mycobacterium tuberculosis. So whether you came down with full-blown TB or not was all about your host genetics. Whereas, so they thought of it as an inherited disease. Whereas now, very few people are exposed to tuberculosis. So essentially all the variation in whether you get it or not is environmental exposure, and we think of it as an environmental disease. But at the systems level, nothing's changed. It's all about where the variance components are.
Yes. So I had a question. You talked about lifestyle, how it can affect these microbiomes. Is there any studies about electronics? Can we use electronics as therapeutics?
I didn't catch the start of that question. Was it basically, have there been studies about how individual therapeutics
Basically,
modify?
if there's any effect with electronics on these microbiomes, like if I play a voltage on it, will it affect it in some way?
That hasn't been done in the human microbiome. So Derek Lovely at UMass Amherst has done some very interesting work in the soil, primarily looking at long-term networks of long-term conductive networks that can extend over many meters in the soil and carry electric charge and modify redox potential over a span that's much larger than the length of a microbe. Whether such networks exist in the gut is completely unknown. It wouldn't be crazy to look.
Okay, and then I had another question was, you guys talk about topical creams to solve these. Can you guys look at like transdermal drug delivery? Using the microbiomes as well?
Yeah, that's something we're actively working on because certain organisms can temporarily change the permeability, tight junctions in the skin, and open them up and have it seal up behind it. So it's basically a microneedle out of a microbe.
Okay, thank you.
I would have also a cautionary statement out of that as a commercial guy. If you start showing that you are absorbing certain compounds, then you're going to do PK, you're going to do safety, and you're going to do a lot of other work. So be careful of what you ask for.
I had a question. So I know, I can't remember if this is one of the diseases that came up earlier on the screen. So right now, I guess my lab is, I'm studying Alzheimer's disease, and I know that the NIH has sort of decided that amyloid beta is sort of a red herring that no one wants to look at anymore. But the other day I was reading a New York Times study that found that exposure to certain microbes might be causation of Alzheimer's disease. So I'm not sure if any of your research or researchers have sort of looked at that connection.
We've done a little work on that. So far the evidence base for bacteria is very weak, but there has been a fair amount of speculation about particular bacteria that might mimic the shape of human proteins in order to trigger Alzheimer's. There's a somewhat better evidence base on viral triggers, which I think is what the New York Times article was about. Although again, you're looking at very preliminary studies and their association rather than getting at mechanism. So I'd say it's intriguing, but not. There's a lot of research to do before the association's proven, let alone anything about causality.
So I had a question about in the first talk early, there was a picture of an office and there was a man in the office, and you said that he had certain bacteria on his skin because of exposure to ocean water. And I was just wondering whether you see either a protective effect or a disease-inducing effect of exposure to different outside environments, whether they be soil or water or some other environment.
Yeah, that's a great question. So most of the data is on that has been on exposure to farm animals and farm materials. Although there's been some work on house dust showing that a more diverse dust microbiome in your house is protective against asthma and allergies. The data on cows mostly comes from Erika von Mutius's group at the Children's Hospital in Munich, where in Bavaria you have farm and non-farm families. where at the individual household level, you see a difference in asthma and allergy incidence depending on whether the family has a cow that they let inside in the winter, and access to hay that the cow has been eating is also sufficient for protection. And then they recently, together with Jack Gilbert, published in the New England Journal last year a study of Hutterite versus Amish families, where some of the differences in hygiene and other cultural practices, even in these genetically very similar populations, lead to large differences in asthma and allergy phenotypes. In terms of soil exposure, there's some data on mycobacteria and their immunoregulatory properties, but there's relatively limited data on soil. There's essentially nothing at all on exposure to seawater, although there's certainly reasons to believe that it might be beneficial in the natural state, as it were, and there's of course great evidence that it's a bad idea to have exposure to it if it's full of fecal coliform and raw sewage, which regrettably is the case if you're looking at the outflows from Tijuana, for example. So that's something that we're just starting to do a lot of work on at UCSD. There's this new Oceans and Health initiative that's joined between the School of Medicine and the Scripps Institution of Oceanography. And there's a lot of work going on at the moment to try to quantify the ocean impacts, in particular, positive and negative, and which of those are mediated by particular chemistry or particular microbiology.
So, I know I said one more, but I'm curious. Anybody got a question for Mark? Commercialization side, product side? Let's do one of these. Okay, she's got it. I saw her at the last second.
Hi, Huda Makla from National University. I have a quick question to run by you. We embed authentic research experiences into the curriculum. We ask students to bring soil samples to culture them on agar and look for zones of inhibition against E. coli and S. aureus. So these halos were really, great halos with zones of inhibition. So how would you take a novel isolate that a student produced in a classroom and then bring it to a product? And to tie it to what the other speakers talked about, do we encourage our kids then to play in the dirt? Because soil sample have these isolates with antimicrobial agents. It makes perfect sense not to be a germophobic. I have 3 kids and I allow them to play in the dirt and get their naive immune system to be educated early on. So any papers on this type of work, and how would you take a student isolate when they jump for joy that they have a positive halo into the pipeline?
I guess I get to start with that one. So if you are able to make a zone of inhibition, then what are you making a zone of inhibition in? And what would be the potential product that you could develop from that? And you could write a patent around that and substantiate that with substantial evidence. And you could file that with the fine folks of Kenobi Martins here, and you could develop a product. That's a lot of work. You could do it.
And in terms of the second part of the question, you should buy the book I just wrote with Jack Gilbert, where the title is literally "Dirt is Good," and it answers the second part of your question in extensive detail, but also in a format that's helpful for busy parents, where you just want to look at the section that's about your question and read that, rather than having to wade through hundreds of pages of narrative to find it.
But I can't emphasize enough as a physician scientist that germs can cause disease, and the hygiene hypothesis for promoting health, there's lots of good association evidence for that, but little specific causation. Whereas in terms of, there's great causation evidence for certain pathogens that can cause disease. And when you're mucking around in the mud, you don't know if you're coming across good guys or bad guys.
Yeah, so you want to pick which mud you muck around in. Although what you say about causation for beneficial microbes was true 2 years ago, but is much less true now. There's been a huge amount of really compelling mechanistic data, especially in preclinical models.
And I would not leave it there without mentioning the hypothesis again, the cleanliness hypothesis that Rich has had in the paper before. You can overwash your skin cells that make antimicrobial peptides are important in defending you. And you can get carried away also and ruin your body's defensive system as well.
Yeah, one thing to keep in mind is the original iteration of the hygiene hypothesis was the idea that you should be exposed to low doses of pathogens, and that hasn't held up at all. Like, there's no evidence that the pathogens per se are important, and you should certainly, you should certainly get all your vaccines and so forth. Like the modern iteration of the hygiene hypothesis is much more about all the other microbes that are harmless or beneficial, not about the pathogens per se.
Okay, and thank you so much, panel. With that, I'd like to thank the speakers again.